response of cells in tooth development.
So, who here ate breakfast this morning? Raise your hand if you ate breakfast. Jason, what did you have for breakfast?
Chocolate croissant.
Chocolate croissant. And how did you eat your chocolate croissant?
I chewed it.
What did you chew it with?
My teeth.
Your teeth, of course you did. So, everyone has teeth, or hopefully you still have your teeth. But we're actually kind of unsure. We kind of take for granted how our teeth develop. And so, in our project, what we're looking at is a process called Wnt signaling. And Wnt signaling may or may not be involved in tooth development. So what our project was to look at Wnt signaling and how it's involved in tooth development.
Okay. So right now in science, people don't really know what Wnt signaling does. They speculate that it can either help cell proliferation, which is when your body grows more cells, or cell differentiation, which is when your cells take on specialized functions. So by tracing which cells respond to Wnt signaling, we can perform further research to see Wnt signaling's true role in tooth development.
So at this point, you may be wondering, what is Wnt signaling? And to be honest, it's a very complicated process. This is just an overview of what it looks like. So I'm not going to go into that, so we're going to give you like the basic summary of what it does. So Wnt signaling creates a protein. It's called beta-catenin, and beta-catenin aids in making more cells, including bone cells.
So the first step of our experiment involves breeding heterozygous mice. So what that is an offspring will have both traits from the mother and the father, and they will both appear in the offspring. So it will have our parent mice, and one will have the gene MTMG, and the other one will have the gene axin-2-Cre. And these combine in the offspring to the genotype axin-2-Cre MTMG. Okay.
So when Wnt signaling occurs, it sends a signal to the axin-2-Cre and it activates it. And once the axin-2-Cre is combined with a specific drug called tamoxifen, there are different processes occur. So what we did in our experiment is on postnatal day 3, we injected our mice with the drug tamoxifen, and when combined with the axin-2-Cre, it causes the MT gene to be cut out. And what's left is the MG gene, or the M green fluorescent protein gene. And that is expressed, which causes the cells to glow green. And those green glowing cells are the ones that are Wnt responsive as well.
Okay, so well we excluded this step, but we're just gonna say it. So basically, we're gonna sacrifice our mice, and that is just killing our mice at postnatal day 5 and postnatal day 9. And this just allows us to see how Wnt signaling migrates in the tooth during stages of tooth development. So then next, we will section our samples, and what this is we'll freeze our samples in OCT, which also stands for optimal cutting temperature. And in room temperature, it's a liquid, and when it's cold, it becomes a solid. So it is very much like freezing an ice cube, but with your sample inside. So then we take our block of sample and put it in our cryosectioning machine. So that's where you put it. And we take a blade and we make very thin slices of this to put on our slides.
Are there any questions up to this point? Yeah.
You said like postnatal day 5 and 9 and things. Why those two and why 2 of them?
So we wanted to see the initial stages of tooth development. So after injecting the mice on the 3rd day after birth, then we wait a couple days, and then we look to see where the Wnt responsive cells are, and then we were looking at the 9th day to just see how it's migrated. They're sort of arbitrary, but we were looking like at the initial stages. Yeah.
Oh, sorry. We were also gonna do a mouse, like look at a mouse in postnatal day 24, but it died, so we couldn't do that. Yeah. So yeah.
Oh, one question is, how did it die?
don't know. You have to ask your twin. But
Complications.
Yeah.
And then I was wondering, you said that it would freeze when it's cold. Do you know around what temperature that
About
is?
negative 20 degrees Celsius.
My question is, why did you use cryosleep instead of
Can you
normal
exit?
cryosleep?
Instead of
Normal paraffin.
paraffin. So the reason we did that is because the green fluorescent protein dies in the paraffin wax.
What is tamoxifen?
It's a type of drug. I don't know exactly its properties, but I know that when combined with the AXIN2-CRE, it causes those processes to, it cuts out that gene we want cut out and then causes the other one to be expressed. Yeah. Okay, so after this we do a stain. It's called DAPI, and what this does is it stains cell DNA or nuclei, and when you view it under UV light, you can see the cells as this beautiful blue color. So the reason we do this is because we want to see the background. So here is like the tooth forming, and then these are all the cells around it. And this here is in the center is dentin, and that's actually made of like calcium, so it doesn't have like live cells. So that's why there aren't cells right there. But these ones around are the live cells.
Okay. Well. Can I like? Oh, sorry guys.
Yeah. Here, we'll just hold on just a second. Here, remove that, and then we'll just show.
Oh, it's back. It's back.
Oh, wait, now it's gone again.
It's back.
She unplugged it, buddy.
Okay, pal.
Hold on.
I'm sorry, family. Sorry for everything.
There you go.
It still doesn't work though.
Okay.
Work on your screen.
No.
No, it doesn't. It's not
Everything
changing.
is broken.
Here, do you want to grab your computer? We have a backup. So it'll be okay. It just doesn't.
Come on, Hannah.
It's like
Use those gerontology skills.
Oh, what?
I think this is not in.
How do you present it usually?
Oh,
There you go.
Okay, it's back. We're back.
Okay. So, the next step, we're gonna do IHC staining, and it stands for immunohistochemistry staining. And what it does is it amplifies the fluorescent signal in our samples. So, well, at first we had a dull GFP, but we wanted to make it brighter. So, we attached a primary antibody. We also attached a secondary antibody with GFP on it. So, and this makes a brighter GFP that can be easily seen under the microscope.
So then we view our slides under the microscope. So first we turn the lights off, and then we use a special wavelength of light. It's 488 nanometers, and the reason we use that is because it's ideal for viewing the green fluorescent protein on our slides under the microscope.
So the results. So what you see here is mass number 7, and it's positive for AXIN2-CRE and also positive for MTMGG. So this was sacrificed at P5 again. So here you see in the green picture that the signal was most prominent in the pulp. And as you remember, this is just a normal microscopic picture, and this just shows like the morphology of the tooth, and this is a DAPI picture.
And similarly with
Point out where the tooth actually is.
Here, like this.
That whole thing is the tooth.
Yeah, but this is the crown, and this is the rim.
again, it's fine.
This was our other mice that we harvested at postnatal day 9, and we're mostly focusing here on the green fluorescent protein. So you can see how the signal is most prominent here, and we've zoomed in, and what this actually is the root. So before it was in the pulp, and so we've seen that over 4 days, the Wnt responsive cells have migrated to the root area.
So what Audrey said, it migrated from the pulp to the root area. So what this shows is Wnt signaling may participate in tooth development from P3 to postnatal day 3 to postnatal day 4.
Okay, so what can we conclude from?
So our project was pretty exploratory, since Wnt signaling is still like a pretty complicated process, and we're not exactly sure how it responds or how it affects tooth development. So what we'd like to do is improve on our project and do some further research. So first, what we'd like to do is do it over a longer period of time. So we only harvested samples at P5 and P9, or postnatal day 5 and 9, and ideally we'd like to look at them at 14, 21, 25 days after the birth to see how the Wnt responsive cells migrate over the course of time. Also, we only harvested one mouse at each harvesting day, and we think it'd be beneficial to harvest multiple mice on the same day to just see if the results are consistent in each different mouse. And thirdly, we would like to look at what would happen if we just got rid of Wnt signaling altogether. Would the tooth even develop without it? Is it critical for tooth development? Furthermore, we'd also like to see what would happen if we overexpress the Wnt signaling, like would the teeth grow faster? We don't really know. So those are just some things we're looking to do in the future. So if you have any questions about our project, feel free to ask. So that's the end of our presentation. Thank you.